You gave me your talk on this. Okay. So first of all, we're going to have some talks by students today. And the first person who will speak is Allison Hardy. And Allison showed up in the lab as an undergraduate when she first took invertebrate zoology, and then she took parasitology, and then she took field parasitology. And now she's working in the lab as a research technician. She, I think, is going to go on to a master's in public health perhaps next year. I'm not sure what her timing is. So she's done some work on some of the parasites of hummingbirds and also some of the parasites of swifts. So we have, the reason we went into swifts because we were not successful in finding very many parasites in the hummingbirds. We had a collection of about 45 hummingbirds from New Mexico at the time. And all they had in them were insects. So they were just packed full of insects, but no parasites in the ones that we actually happened to look at. And so we said, well, let's look at something that's close. So we looked at swifts because it's in the same group, the Potiformes. So let's go ahead and start the PowerPoint. I can do that here. Work. And I probably have to do a share again, eh, Gabor? Yeah. Okay. In Zoom. Okay, start the PowerPoint first. There we go. Alt tab. Oh, you have to hold it. And then we. Go to share. And then I'll share this one. It should work. Windows 11 is slightly different than Windows 10, so it's one of the problems. I think this is ready to roll there, Allison. So this is, come on up. Allison is really working hard on this project. We, when I was in Mongolia, I was up on the top of this, up above a Pleistocene lake bed. And as I was going up the side of the hill, I was getting snails out at different layers. And they looked exactly like the snails we get around here. Very interesting assemblage of snails. When I got to the top, these swifts were flying all around. And these are swifts. They were flying around. I happened to have a 410 shotgun, and we were collecting these things. So every time one went by, I shot at it. I thought, oh, I was missing because they just went off the edge and I couldn't see over the edge. It was so steep. Augie came up and he had 8 of them in his hands. He said, you haven't missed once. So I got a good collection of swifts from this area. And so that's what Allison is going to talk about today, were those ones that I actually collected in Mongolia, mostly from this one locality. And it was an amazing locality because we also got terracles in the same spot, which are sand grouse, if anybody knows that sand grouse is very interesting groups. So this is Allison's work. We're working together. It's a pretty interesting group. So thanks, Allison.
Hi, everyone. Thank you all for being here today. I am, like Scott said, going to talk about some parasites of the Potiformes and then also some insects and and some arthropods associated with them. So, so a little bit of background on Potiformes. It's traditionally made up of 3 families: the hummingbirds, the swifts, and the tree swifts. There's some debate among taxonomists about whether or not Apodiformes should be raised to a superorder, and hummingbirds should become its own order separate from swifts, but that is still being debated. Regardless of what they decide, hummingbirds and swifts are still each other's closest related groups. So they're a very fascinating group of birds to look at. Hummingbirds have 366 species, making them the most abundant group of apodiformes, but they are only found in the New World. Nowhere else can you find a hummingbird. They range all the way from Alaska down to the tip of South America, and that's where most species are concentrated, is in Central and South America. The swifts have 113 species, distributed worldwide, except for extreme northern latitudes and some islands. Here in the North America, they're not as commonly seen. We only have 4 species, so the majority are found in Europe. And tree swifts have only 4 species belonging to a single genus, and they're found only in India and Southeast Asia. So hummingbirds are really interesting in terms of the lack of knowledge of their evolutionary timeline. Some ancestral hummingbird fossils have been found in Europe, majority in and around Germany. These fossils these fossils were also nectivores capable of hovering flight, just like modern day hummingbirds. The photo that you can see up there, that fossil is estimated to be around 30 million years old, 30 to 35 million years old. And we don't know much about the time in which they came from Europe over here. The spread is not well documented, mainly because of the lack of fossil evidence in North America. We only have hummingbird fossils as old as 10,000 years ago, and they are believed to have begun diversifying in America in around 22 million years ago. So there's a big gap in knowledge, and we don't know how they got here either, because the proposed methods for which they came over here, either the Bering land bridge or transatlantic flight, neither are well supported. Most people don't believe that hummingbirds could have made it across the Atlantic Ocean without stopping. And because there's no fossil evidence anywhere in North America to provide a record from very north in Alaska all the way down to where they're at, or where they're found now. So it's still a mystery, but it is one of the things that makes this group so interesting. Another thing that makes hummingbirds so interesting is the lack of parasites that have been identified from them. So this is everything from all 366 species. These are the only records of parasites, and the majority of them are in Central and South America. There are 4 species of tapeworms, 2 of trematodes, and 1 nematode that have been described from hummingbirds. Before 2019, no endoparasites had ever been reported from hummingbirds in North America. Several cestodes were found during a histopathological study in California, but no one tried to identify them. So they just said there were cestodes, and that's all we know. Earlier this year, we got really lucky, and there was another record from Mexico, only to genus level, but still really interesting because that's the first named parasite we have from hummingbirds in North America. But with this severe lack of knowledge of these parasites, we were really interested in looking to see if we could find any more. And so that's where the museum collection came in. Collecting hummingbirds can be really difficult because of the multiple permits required to collect them. Even just salvage hummingbirds off the ground requires permits. So we were lucky that there were some preserved hummingbirds in the Mantor lab that we could use for this project. 41 vials of preserved hummingbird digestive tracts were located in the collection. They were collected mainly near Albuquerque between 1987 and 1989. The map shown, the number in each dot is the amount or the number of individual hummingbirds that was collected at each location. When we went to examine them, 14 of the vials had become completely bone dry and we were unable to look at them, but the remaining 27 helminths were screened. These are the 3 species that we looked at. The black-chinned hummingbird, which has its breeding range in New Mexico. The rufous hummingbird, which migrates through New Mexico to its more northern breeding range. And the broad-tailed hummingbird, which is found at higher elevations in New Mexico during the breeding season. Unfortunately, we did not find any parasites, lots of work and very little reward, but we did find some arthropods in the hummingbird digestive tracts that had not passed through the stomach yet. So they were mostly whole, missing a few appendages like wings and legs, but we were able to identify most of them to family level and 1 to species, which was the black flies, species Simulium vittatum. Additionally, we found biting midges, non-biting midges, leafhoppers, wasps, and those all came from the black-chinned hummingbird. And then we found an orb weaver spider in the broad-tailed hummingbird. Currently, this is very exciting, we have some hummingbird cestodes and nematodes that were sent to the lab from Arizona that we are currently working on identifying. They came from different species to what we looked at in New Mexico, and they're mainly window kills around Midwestern University campus that are being picked up and then screened for parasites. So when we get them, they're not in the best condition, but we've been able to identify 3 different genera, each already known to infect hummingbirds, but only one of those has been found in North America so far. So it's really exciting. And we don't know species yet, but we're going to keep working on that and hopefully we can figure out the rest. So when, as Scott mentioned earlier, when no parasites were found in the hummingbirds, we looked back into the collection to see what else we could look at. And we had some other apodiform parasites that were collected in Mongolia, which very far away from New Mexico, but it was the closest we had to what we looked at in the hummingbirds. And so we got parasites. So 11 hummingbirds were collected from 7 different localities in southern and southwestern Mongolia between 2009 and 2012 as part of the Mongolian vertebrate parasite project. I have a few photos to show from the areas in which these swifts were collected. So this shows just some of the habitat diversity in southern and southwestern Mongolia. Some really interesting habitats. These cliffs could likely be homes to swifts because they typically make their nest on the faces of cliffs. That's their preferred habitat. There's some grasslands and some forested steppes, which are all very, very cool. So the species that were collected, there are three species of swifts found in Mongolia. We collected 2 of them. So the common swift, Apis apis, which is spread all throughout Europe. It's a very abundant bird, but they are very long distance migrants. They travel all the way to sub-Saharan Africa and then back up to Mongolia, which it's an incredible distance that they fly. We also collected the Pacific swift, Apis pacificus, which breeds in Mongolia also, but it winters in Southeast Asia and Australia. So they meet up in Mongolia and then they go their separate ways during the winter. So knowledge of bird parasites in Mongolia is also very limited. Only one major endoparasitic survey was done in 1964, and no Swifts were collected. And so there's really no record for these birds in Mongolia. Some parasites have been collected from other countries in their ranges, but nothing in Mongolia. 12 cestodes, 12 trematodes, and 7 nematodes have been identified from these Swifts. And we found that 7 of the 11 Swifts we looked at were infected with some of these known parasites. The cestodes and trematodes that we recovered were stained with either carmine or hematoxylin. They were mounted in gum damar and measured using a Zeiss axiothot microscope. We based the identifications mainly on morphology, but we also compared them to the known ranges of measurement values for the species that are in Swifts. And now to the fun pictures that we found. So this is Pseudangularia europea. It is a dilopidid tapeworm found, described in Hungary from the common swift, and it has only had one other published record from Ukraine, also from the common swift. It's a really interesting tapeworm. This one was stained in hematoxylin. We found it in Apis apis and Apis pacificus. So it has never been reported from another host besides Apis apis. So this is a really interesting finding. And also, we only had 7 infected birds. This parasite was found in every single one of them. The Apis apis each had a single pseudangularia. The Apis pacificus had 14. So a really big difference in the intensity of the infections of these different species. It was in the small intestine when it was recovered, and it is a very, very cool thing that we found, I think. This is a photo of the mature segment of it. This is from the original description from Hungary, and then hopefully you can see everything well. I'm kind of getting a glare from the window, but okay, good. The top photo shows the female organs, and the bottom shows the male organs of the tapeworm. And I believe my photo of the female organs are in focus there. It's a really interesting tapeworm in that it has this really thick cellular sheath around the female organs that is I mean, it's very prominent, a very prominent characteristic. It also has something that is not found in many tapeworms. It's only found in 3 genera of dilopidid tapeworms. It has this, it's called a vaginal sclerite. It looks like a little dumbbell almost that is, oh yeah. Laser point. Okay, thank you. Sorry. So hopefully you can see right here, it kind of looks like a little dumbbell shape, and that's a very rare characteristic for tapeworms. We also found Notopentorchis vesiculigera. This is a paruterine tapeworm. It was described from Apis apis in Denmark, but it is widespread and in lots of different species of swifts at temperate latitudes. We found it in 3 of the 11 tapeworms we looked at, all apis apis, and it had an intensity of infection from 1 to 6 individuals per bird that we looked at. Oh, additionally, sorry, going back, Pseudangularia has 3 rows of hooks, or it's a single row of hooks that zigzags almost to make it appear. Oh, sorry. It has 3 rows of hooks that kind of go in a zigzag pattern, and then compared to Notopentorchis, which has a double row of hooks, all of which are the same size in 2 separate rows. This is the mature segment of Notopentorchis. They're hard to identify a little bit. The parts are kind of hard to see. So this is just showing that some of the difficulty when it came to measuring. We had a hard time finding some of the structures that we needed to, but hopefully this is one of my best photos for showing some of the internal structures. And then those were the 2 cestodes we found. We also found 2 trematodes. So Brachydistomum olsoni is a dichrocelid trematode. It is very common in swifts across Europe. A recent study surveyed 250 swifts and found over 800 of these inside of them. So they're very prominent. They are parasites of the gallbladder, and we found it in just a single swift, and there were 6 of them. We also had Plagiorchis maculosus, which this is a more frequent parasite of swallows than it is of swifts, but it is well known and well described or well documented throughout Europe. We found it in the common swift again. There was only one in a single bird. There was just one of these, and it was in the small intestine, which is, yeah, sorry. I apologize. I ran through that all very, very fast, I think. But I would like to thank the NSF, the Harold Manter Lab, Scott and Gabor for all of their help with this project. And then William Baltasar and Jason Struthers for collecting the hummingbirds that we looked at, both in New Mexico and Arizona. And also, if anyone has any questions, I would love to answer some. Yes.
Bring the whole bird back
So the swifts were processed in the field following standard protocols, and then just vials of the parasites were brought back. So that's what, when we looked at the lab, I just had lots of vials, and then I had to stain them and mount them, and then look back to see what they came from. Yes.
Were you ever able to get in touch with
I was not.
We did collect
Okay. Okay. Yes, I had your record from the trematode. Sorry.
Right. Yeah.
Oh. Yes. Yes.
But I know we got it.
Okay.
All the birds ended up, going to.
Yes.
They haven't published against that.
Yes. Yes, it should. I couldn't find any published record of it, but yes, I should definitely reach out. Yes.
probably not the same one. But
That would be great to, yes.
And I know, my memory is that, we didn't keep close track, my memory is that those birds we found a lot of white.
Oh, yeah.
The article called Black One.
Yeah. I think, I mean, we ended up sort of deciding, after looking at all the, what I did, they were mostly, what they were doing down in those flowers.
Mm-hmm.
They were mostly going after, they got
Yeah.
the nectar as byproduct,
Yeah.
but it wasn't, so the whole co-evolutionary story about sequence flowers and stuff like that may have more to do with insects hiding in the flowers
Yeah.
than the nectar may on.
Yeah.
Especially since, you know, you get one of us that were down in the drive, there are flowers.
Hummingbirds definitely rely on,
Numbers are there.
yeah, on the insects. Yes. Oh, good. Yeah. Yeah. Yes, it is. Yes. I would definitely be interested in looking to see if any other museum collections have specimens that I could look at from hummingbirds. Right now, the hummingbirds we're getting from, or the parasites we're getting from Arizona, that has been great to have just extra information on these things. So I would definitely be interested in looking to see what else in North America is out there.
So unfortunately, especially coming from Arizona, are window kills, and so the big ones are rotten by the time we really need to go out and collect a whole bunch of them. Instead of trash, instead of that one.
Yeah, but so yeah, the majority of those window kills have been not in the best shape, but what we have been able to look at has been, some of it has been very useful. So
The problem with those is that hummingbirds, once they get killed in a window in Arizona, then they heat up more than when they were dead.
Yes.
When they were alive, so they heat up there.
Yeah.
Any other questions? Thank you very much.
yeah, thank you.
All right, so who's next? Does anybody remember the schedule? You're next, okay. So bring up, let's see. You don't? I do. Let me give you a flash drive and we can. That's okay. I was trying to put one on a flash drive yesterday, and the problem was my son had put a Linux distribution onto the flash drive. So it was, I said, why is this not working? Because I couldn't see the files because it was Linux and I was using Windows. So I finally figured out what was going on. Hi, Carl.
Hello.
It looks like you're muted, but maybe everyone's muted from out in the wild.
Mm.
We should be able to start learning the dialect of red-winged blackbird soon. Okay. Thank you. anybody notice that when you try to put a USB drive in, almost 90% of the time it's upside down? It seems to me it should just be 50%. Unless you look. And then I can't see, so it doesn't matter. Which one? Oh, let's see. Here we go. Go down here. USB. Okay. Yeah, it should be there. Oh. This one?
Yeah.
There we go.
Should be right.
It's opening.
Oh, there we go.
It's so giant.
To be honest, I don't know how it goes now, but no.
Okay. Thank you. I didn't know. Did
There's
it-
actual Hungarian food, I was definitely wrong.
Yes, we'll have to ask them. Well, what about- yeah, you could ask about what. That's good. So I probably have to share again, Gabor?
Yeah.
And I said I'm supposed to hit alternate tab. And then I go to Zoom where Carl is. Then I go to share. And then I go to here and say share. Good thing you're here, Gabor. All right. So the next undergraduate, are you still an undergrad?
Yes.
The next undergraduate, see, Allison's not quite an undergrad, she just graduated, but Charlotte is an undergraduate. So she's going to speak about comprehensive and efficient workflow for pathogen screening, data capture, and vouchering of insect vectors. So Charlotte Sakaris, how do you say that in Hungarian, Gabor? Sakaris? Okay, so I didn't do it right. I'm sorry.
I didn't do it right.
So this should be running. You can take it away. If you want to use the little pen thing here, I'll just start it for you so we can do laser pointer. And this is a little bit easier. It's less sensitive. You
Yeah,
wanna
sure.
use a laser.
And then that disconnects.
Yeah, and then for next slide, we'll go down here like this. And then you can use this from then on.
Okay,
So
cool.
you don't have to mess with the mouse.
Thank you so much. All right, thank you for having me. It's an honor to be here. My presentation today is on a comprehensive and efficient workflow for pathogen screening, data capture, and vouchering of insect vectors. Just to provide a brief outline, I'll first be introducing the project here. Then I'll be explaining what phytoplasmas are, why it's important to study them. I'll give an overview of the workflow used in this project, the results from the data thus far, and then I'll be concluding. All right, so this project is a large-scale project with a number of general objectives. These goals overall are to study the biodiversity of phytoplasmas, insects from the order Hemiptera. We're mainly focusing on leafhoppers and treehoppers, and their host plants. And also a goal is to study the tritrophic interactions that occur between these three different groups, and then use this information to predict the emergence of infectious plant diseases. The procedure described in this presentation has more specific goals that will go on to contribute to the overall goals of this project. Procedural goals described here are to collect specimens from around the world, to collect a diverse selection of samples to test, recording the information from these specimens digitally for easy access and efficient organization, screening these samples for phytoplasma in order to document the relationships between phytoplasma and vector, and vouchering these specimens for future reference. Just to give an explanation of what exactly these tritrophic interactions are, I'll be describing the three different interactions. The first one being the interaction between the insect and the plant. The second interaction is between the phytoplasma and the plant, and often this can be asymptomatic, but sometimes it displays itself in ways such as what you can see on the screen here, where a normal echinacea plant instead grows these leafy growths. And then there's all sorts of other different symptoms that plants can show when they're infected by phytoplasmas. And the third interaction is that between phytoplasmas and the insects that they infect. What exactly is a phytoplasma? It's a type of bacteria that lacks a cell wall, and it's often present in plant phloem, which is where leafhoppers and other hemipteran insects often feed from. And it can cause diseases in plants, and it can also be asymptomatic, which is what makes it really difficult to tell whether phytoplasma is present just by looking at the plant. It often needs to have DNA extracted, and then you do all sorts of processing to figure out whether the phytoplasma is actually present. And another part of what makes this type of bacteria really difficult to study is the fact that it's non-culturable, and it thus must be studied in vivo within living tissue. And then just to show what they look like, here's an electron microscope image of phytoplasma, which can be seen as these little round shapes here. And then the image on the bottom is showing lethal yellowing disease, which is currently a big threat to palm trees. Why is it studied? Why is it important to study phytoplasma? Currently, a lot of effort is going towards studying phytoplasma because of the massive amount of economic loss it causes, especially to economically important crops. And the research is focusing mainly on mitigating the damage caused by phytoplasma diseases on these economically important crops. It's important to study further because if we just study to understand how phytoplasmas interact with plants and insect vectors, we can start to understand the underlying mechanisms of infection, which therefore helps us understand how these tritrophic interactions work and come about. And this can help us make predictions and take proactive measures against plant diseases in the future rather than just reacting to them when they occur. So I'd like to give just an outline of how phytoplasma infection works. It starts when an insect feeds on the phloem of the plant, which contains phytoplasma, and this is called acquisition feeding. From here on, the leafhopper is virulliferous, meaning that it carries the phytoplasma, but it cannot necessarily transmit it yet. There's a period between which the phytoplasma enters the body and when the insect becomes infectious, and this is called the latent period. And during this time, the phytoplasma multiplies within the body of the insect. At this time, systemic infection begins, which requires passing two major epithelial barriers within the insect, which when passed, mean that the insect is then capable of transmitting the phytoplasma to other plants. It first must, when the insect first feeds on the plant and obtains the plant tissue and the phytoplasma enter the gut of the insect. From here, it must pass the epithelial barrier between the gut and the hemolymph, and then back from the hemolymph into the salivary gland. And at this point, when the salivary glands are infected and the insect goes to feed on another plant, this is called inoculation feeding. The phytoplasma from the salivary glands of the insect is able to enter the plant, and this causes further infection. And then the cycle continues again. This is from understanding the various steps that are required for disease transmission. We can begin to understand the relationship between the phytoplasmas, the insect vectors, and their host plants. This is an overview of the workflow that was used, or that I did when contributing to this project this summer. We start first by extracting the DNA from insect specimens, then we screen them for phytoplasma, and then use next-gen sequencing to obtain their genetic sequence. And through this process, we can obtain the insect species, the type of phytoplasma they carry, as well as the type of plant that they feed on. Making these associations is really crucial for predicting and possibly preventing the spread of phytoplasma, as well as documenting biodiversity, which is one of the main goals of the project. First things first, I want to mention the collection database that we use for digitization throughout this process, which is called PaxonWorks. After nearly every step in the process, we digitize the information, and this is really essential for the vouchering process, as well as keeping information regarding every single sample organized and easily accessible for future reference. The information that we digitize consists of the collection information. So this is longitude, latitude, altitude, the method of collection, and multiple other important details. Then we have individual specimen information, which is going to include the taxonomic classification, as well as like the individual insect code so that we can go back and refer to it later. And then it also includes DNA information from after the DNA has been extracted. All right, so we first start off with a bulk sample. The bulk samples addressed in this presentation were drawn from a large collection of samples that have been collected over a long period of time from 1989 through 2022, and it's collected from countries all over the world. And due to this wide range of collection dates and location, we're able to look at both the temporal data and the location-based data. And just to provide an idea of how widespread the collection was, this map shows all of the points that were analyzed for this presentation, all of the positive samples, all of the samples that have tested positively for phytoplasma. And as you can see, it's across a wide range of countries, a wide range of different climates. So we have a pretty diverse selection of insects here that were analyzed. And for the actual process of going through the bulk sample, we first start out with the bulk vial, which is collected through some method, and then the collection event is entered into the database. And we start out by splitting the vials into morphospecies, which are basically just species that look the same, have the same or similar morphological characteristics. And then the morphospecies are split into separate vials. All right. And then I have some images just depicting the previous steps. Here is me using light collection to collect insects in Illinois. Here is an example of a bulk vial, also from that same collection. And then this is what it looks like after you've split the bulk vial into morphospecies. And then those morphospecies, each one of those little piles on the tray, are given their own individual vial. From here, we proceed to the single sample after identifications have been made for each of the morphospecies. We take the morphospecies vials, and then we ideally take 4 individuals from that vial, 2 males and 2 females if it's available. If there's less than that, then we just take whatever is in the morphospecies vial. Each individual is entered into the database, and then the vials are ready to proceed to DNA extraction. And then here is an image of 4 vials, 2 males, 2 females, and they're all of the same morphospecies. So each one of these individual vials will have its DNA extracted in the next step. All right, and now we proceed to DNA extraction. We start with the the individual vials, each with 1 insect. And the individual specimen is washed in bleach in order to remove any debris and genetic material from the outside of the insect. The insect is then rinsed in water to remove the bleach, and then the abdomen is removed in order to release more DNA during incubation. And then test buffer and proteinase K are added in order to extract the DNA from the insect. Here I am rinsing the bleach from the insects and then removing the abdomen. And then the photo on the right is just an image of one of the insects with its abdomen removed, showing how it really just opens up so that the test buffer and proteinase K can get in there and extract as much of the DNA as possible. The second part of the DNA extraction begins by removing the insect's body, and this this body is set aside so that it can be vouchered later and referenced, and I'll mention that later. And then the DNA, which is still present in the liquid, is purified by adding helix resin, which binds to all the positively charged debris present in the vial and leaves the pure negatively charged DNA just present in the liquid above. And then at this point, the purified DNA is ready for qPCR, which we'll go over later. I just want to give a brief overview of vouchering and its importance. Vouchering is the preservation of a physical and digital record of each specimen that we extract the DNA from. The digitalized information is present in the database, and then the insect's body is pinned with a label with the collection event information, but also with a little QR code so that you can access the digitized information easily just from the physical specimen. And then after the physical specimen is mounted, it's mounted in a box with hundreds of others, and then these are all put into storage for future reference. And the importance of vouchering is that it allows us to be able to refer back to specimens at a future date, and it allows us to organize all the information about these samples so that we're able to reference it when needed. And it's also especially important and helpful when revisiting taxonomic classification in the future. All right, so if you remember the DNA that we extracted in the extraction step, it's now proceeding on to the qPCR step. And the qPCR screening works by amplifying leafhopper DNA. And because it's quantitative, we are able to quantify the leafhopper DNA versus the phytoplasma DNA and generate a CQ value. In this project, a CQ value of less than or equal to 32 is considered positive for phytoplasma. There is a bit of a gray area, which I'll explain later. And a lower CQ value means more phytoplasma is present in the sample, since it took less time to reach a certain threshold of phytoplasma DNA. The quantity of the phytoplasma and thus the CQ value can suggest that the insect was a carrier of phytoplasma. However, it's a lot more difficult to determine whether an insect is capable of transmitting DNA or transmitting phytoplasma, as there's numerous factors at play here. For instance, the insect could have just eaten the plant and then died without having time for the phytoplasma to travel to the rest of the body. The phytoplasma could be just present in the stomach, meaning that it's incapable of reaching the rest of the insect's body. And for this reason, actually, some studies will separate the body from separate different parts of the insect's body and analyze them separately. So that they can tell if it was capable of reaching the rest of the body, and thus if the insect was capable of transmitting phytoplasma. And this is the kind of qPCR graph that we use to determine whether the samples were positive or not. All right. And at a certain point, a curve, after the phytoplasma DNA has reached a certain concentration, a curve will be generated. And this I just included to show the distribution of CQ values for all the positive samples that were analyzed for this presentation, just to show the spread of values, how most positives fall at around a CQ value of 32, and also to show that how even samples with much higher CQ values can test positive. Just to reiterate that the CQ value is not a certainty, it's more of a suggestion. And from then, after we've determined which samples are positive, we can send off the DNA samples for anchored hybrid enrichment, or AHE, which is a form of next-gen sequencing. This sequencing is a type of sequencing that produces, after some processing and reconstruction, a genetic sequence of specific portions of the extracted DNA. And from these portions that we receive the genetic sequence for, we are able to tell if phytoplasma is actually present in the insect. We can identify the phytoplasma strain, the plant that the insect consumed from the tissue inside the body, and then we can also make more specific insect classifications and help arrange the insects phylogenetically. And this all helps us draw a much better picture of how these tritrophic interactions work. So the results that are analyzed here are 148 positive samples that have come from a much larger collection of data of 2,300 total samples. So as you can see, not many end up testing positive. It's a very low positive rate. Many of the samples that were analyzed here had not yet been fully processed via AHE, and therefore the plant associations and phytoplasma association, or the more specific plant associations and phytoplasma associations, were not available. Therefore, for analysis here, I used just plant associations that were found via the literature. And I also analyzed the information that had been digitized, such as collection event information and taxonomy. So here you can see the frequency of the various classes of host plant that I found in the literature. The most common host plant being grasses, followed by forbs and woody plants, and then a smaller number of species fed on a mixture of two or more of the other plant types. And this here is the same data, except also considering the region that it was, the sample was collected in. So of the many, many countries that these samples originated from, I grouped them into 4 different regions: America, Africa, Australia, and then Europe and Asia. So from this, we're able to see that in Europe and Asia, the species that typically consume forbs are most commonly positive at 25. of the samples analyzed here. And then in the Americas, species that consume grass are more commonly positive at 26 positive samples from the samples analyzed. And from this, we can draw a few different conclusions. As these plant types in these specific regions seem to harbor more phytoplasma positive insects, it would be best to focus our preventative efforts for infectious diseases in these areas. This is definitely much more of a general overview though, and once more information is available, such as specific plant species or the associated phytoplasma strains, we can be even more targeted in our efforts to prevent or be proactive about emerging infectious diseases in plants. Overall, this organized workflow developed for this project is essential for completing the project's overall goals, which are documenting biodiversity as well as tritrophic interactions and preventing or being proactive concerning the emergence of infectious diseases. The collection, vouchering, and databasing, as well as next-gen sequencing, are all extremely important for making these associations. This approach does have some inherent limitations, not really as a fault of the procedure itself, but rather just by virtue of the subject of study. And this is because we can know that the phytoplasma is inside of the insect, so we can make the phytoplasma insect association. We know that the plant is inside the insect, so we can make a plant insect association. However, we do not know if the phytoplasma has been acquired via the plant that is found inside the insect or a different plant that was consumed prior. The insect may have fed on numerous plants. We don't know which one the phytoplasma is from. So we can't really for certain draw that connection between the phytoplasma and between the plant. All we can do really is make inferences. So this workflow maintains organization through digitization at each step and databasing to keep all of the samples and associated information readily accessible. It would be impossible to use this data to predict the emergence of infectious diseases and to make these associations and to make these analyses without an efficient workflow that utilizes databasing and vouchering. And this presentation is a part of the REU program that's funded by the National Science Foundation. And I also wanted to give thanks to my mentors, Valeria Trivalone and Christopher Dietrich, for their support along the way. Thank you so much.
Really cool stuff. Any questions?
Sorry, wait.
No, that's it. But if it ever, what I'm leading up to is, you get secondary, you get so they don't, once you get infected.
So can you also, can you look at one of those little leaf hoppers or plant hoppers and using DNA tell which species of plants it ate? Yeah. So you can like say, okay, 5 different species of plants.
Any transition that was put in that line of that, we can tell.
Right, and so, is there a certain lower limit to be able to find that out, or is it pretty much see all different plants today?
Well, after it goes through the. Run that through black, and then. Yeah.
It's pretty fast.
Yeah.
So is it more common to find, the stomach content really dominated by one kind of plant, or all of them all?
Yes, so not entirely. Based on the literature. I would bet that there's nothing.
Yeah, see, that's my,
Those are the.
that's my suspicion is, that what we see oftentimes is what the led to effect. So you pull these insects off of those plants, and the assumption is that they're specialists in it.
But yeah.
And, so this kind of work is, I suspect this kind of work is more. All of those assumptions affect general. That's going to be. That's going to be a couple of months. Okay, any other questions? Anything from the internet? Oh, Tom. Private presence, they just have to drain one sign. Or the sign, or the various kinds, and various strains of each kind. Within the plant, any particular individual plant, do you find more than one species, or do you find only one?
I believe the plant could have one and one.
If it has more than one, within leafhopper, is it infected with more than one kind?
It's
At the
possible
same time?
the leafhopper was, that's what was. There's two different areas that have that, and only some metacarpal areas get that.
Are there any cases of, where you found where although it can't be transmitted, the leafhopper continues carrying it?
I believe that if cytoplasma is capable of moving the gut, yeah, if it's not capable of detecting the rest of the body, yeah, it can contribute to the.
That's not always the first strain is required.
To take direct samples from any systematic plan that you find.
So,
Compare the bioprocess found in that plan with the normal bioprocess you found in the insect. Because like, just thinking for the crop perspective, right? I feel like that would be information that you could say that based on our sampling, if you find this many systematic plants in the crop, that means that this is the flora of mycoplasma you're dealing with in the plant. Yeah, I mean, I'm sure that would be effective, extracting DNA from plants, I never seen. I did some in Italy and it took us 4 hours. But I think if it was, I think it's feasible, if you're just focusing on that, on the incorporating the relation, and that's more what we're focusing on here. But I think it was.
Sorry, this is too cool. So, do you have any information about insects preferentially feeding, selecting, or avoiding diseased plants?
I haven't seen anything about it.
There are.
There are.
And information about it. A lot of funds of research that prove that infected plants, they can actually produce a protein receptor that attract more healthy ones, like that. And then you also prove that there are special proteins in the plant that cytoplasma producing plants that make the insects to be more to increase the efficiency of the insects, to lay more eggs and like provide more nutrients for the insects in order for them to lay more eggs.
Probably not going to be
I have another question. Do the insects bite each other and transfer these things back and forth? Do the insects actually bite and transfer things back and forth? Each plant, each individual insect. I know a lot of these biting insects bite each other when they're in a jar.
I never heard about it.
Because you put a bunch of, for instance, vegetable bugs,
Yeah.
and they start poking each other.
So
I'm wondering about.
aggressive behavior,
Yeah.
I'll be honest, I never read anything with some either having this behavior, but I will look at it. Yeah,
Pick them apart.
yeah, but this has happened with the low feeders.
Yeah.
Interesting.
Well,
Maybe they are less,
those fast feeders.
maybe big vegan insects are less aggressive. I don't know.
Yeah, but the thing is that those that are the fast feeding vegetable type things, that don't only eat bugs, also attack each other.
I should,
Attack the bugs.
yeah, I should have a
Maybe
look.
they're predators.
Interesting.
Very interesting. Any other questions? Anything out in the internet land for questions? Is there something? No. Okay. So, anyway, there's a lot of people out there on the internet today, so I'm trying to get this to pick up our questions. I'm not sure if it's working or not, but we'll try it. So any other questions or comments? All right, so basically we have some time now. We're supposed to have a break. We seem to have gone a little fast. So we're supposed to have a break about now. I think 10:30 is fine. And so we're going to start again at 11, but the coffee didn't show up yet. So we'll have to just kind of mill about until 11 o'clock. Then, yes, have them go get some for us. Okay, well, thanks everybody. We're going to be back here at 11. It gives you some time to look around the museum. There's all the way down to the basement. There's all kinds of really interesting displays here. The student union is just over that way. You can go get coffee if you'd like or anything. So thanks for being here, and we'll reconvene at 11 o'clock Central Time. It's now 10:20. So maybe I should write a note to the, could you put a note to the internet people that says that? So Lindsay, do you have, excellent. I'll put it on here then. If you, yeah, I'll come get it.
Yeah, I needed to buy a memory stick anyway, so. No, I'm sending my second chapter tonight. It's done, I just need to edit one of my images. And it's fighting. My image is fighting.
The parasite stuff out there. And so don't feel like you can't just belt it out. Act like you're speaking to a bunch of freshmen when you're speaking here, okay? Remember how that was. Okay, so right now, one of my favorite paleontology students is going to speak about digitization and organization of invertebrate collections, which is another one of my favorite kinds of things. Invertebrates, things that don't have vertebral columns. Various kinds of, I'm actually going to not say what I was going to say, but let's keep, you can imagine what I would say. So we'll go ahead and keep moving here. So Lynn, just next week, she will finish her master's degree here working on ammonites. And she said today or tomorrow, I will get her thesis and I will finish reading it again. And then next week we'll have her defense. And then she's going off to start her PhD program at University of Cincinnati. And she will blast through and come into academia in a way that you can't even imagine. So Lynn, take it away.
All right, so I'm going to talk about things that died 50 million years ago, which is exciting. As he said, I'm a paleontologist, but hello, are you going to work for me? There we go. I got it. We're going to figure it out.
You just use this way and this way. Left
Down?
and right. Left and right.
Oh, left and
Yeah.
right. Perfect. All right. So we're going to start with a brief background about me. That photo over there is me 2 years ago, graduating from the University of Nebraska-Lincoln with an animal science degree. So I'm currently a second-year master's student in the Earth and Atmospheric Sciences Department, which is that building right over there. I work underneath a paleontologist named Peter Wagner. We do invertebrate paleontology. So I study ammonoid phylogenies. So the ammonoids, as you can see on the little background, are the swirly looking fossils. They kind of look like if you took an octopus and shoved it into a snail shell. I also look at diversity and disparity at various taxonomic levels to see, you know, should we be doing research at the species level, or is it okay to do research at the genus or family levels? Because a lot of paleontology, we have so much stuff that we kind of do it at a higher taxonomic level, but we shouldn't. Spoiler alert. As Scott said, I will be attending the University of Cincinnati in the fall. I will be transitioning from metadata research into isotopic research on my specimens, ammonoids. So, undergrad, I started as a pre-vet student. I wanted to be a zoo vet. And I wanted to work with endangered species. I wanted to do genetics work. However, sophomore and junior year, I took a part-time job up in north of town at a UNL pig farm. And I hated it. It was the worst. I felt terrible. I realized, I came to the conclusion I could not euthanize animals, which is a big part of being a vet. You know, if you gotta do it, you gotta do it. And I couldn't. So it's not for me. And I made that realization junior year of undergrad. I had already waited to go to school, so I felt this big time crunch. What am I gonna do with my life? I've already spent 3 years with this dream. Am I stuck with it? What am I going to do? During that same time, I took an intro to paleontology class in fall of 2019 under Pete Wagner. I spent that entire semester harassing this poor man. I approached him multiple times. I emailed him papers. I asked him about his research. I just kept harassing him. And then finally, he turned to me at the end of the semester and he went, "What do you want? Do you want a job? Like, what do you want from me?" And I was like, "A job would be great. That'd be awesome. Thank you." So senior year of undergrad, I started assisting his graduate students and his wife, Kate Lyons, graduate students in data collection. So I was the one that had to read all of the papers, pull out the important data, and send it on to the people who were doing the fun research. During that time, I was also asked to set up and start the digitization efforts in our invertebrate collections with the current emeritus curator, Bob Diffendahl. So we're going to leave Morrill Hall and take a tour through campus to our collections. So we're going to start Morrill Hall. This is where we are now. It's hot outside, so we're just going to stay in here. We're going to hang a right and start heading towards the Howard School of Business, no relation. We're going to pass by the School of Business through this beautiful little grassy area, hang a left, and pass by the School of Education. Sorry about the photo bomber. We're going to do a little shortcut because, you know, I'm mosey, I walk slowly. We got to find some ways to get there faster. So we're going to cut through this parking lot, past the School of Physics, also past the School of Physics. And here we are at Nebraska Hall, which I was under the impression was the School of Engineering. Apparently they moved. I didn't know that. So we're going to enter in through this door, go down these creepy, creepy stairs, hang a left, and go through this hallway that allegedly was full of asbestos when I was working there, so that's cool. And we're gonna stop at this door. It's, you know, not very dramatic, but inside that are our invertebrate collections. We have 7.5 million specimens spread across 2 rooms. We have multiple drawers and cabinets that we have organized by rock formation and then age. So we have a member and then the formation and then the overall age. And most of our specimens were collected before 1960. So they're not only are they old in the sense of 100 million years, but they're also old in the sense of getting dug up from the ground. And many did not have genuine specimen tags. I did this during undergrad, so unfortunately I didn't take a photo of this tag. But at one point, I found a specimen that had a tag next to it written on the back of a piece of newspaper. So it's like a round type print and stuff, and it said 50 yards south of the large oak tree on Mrs. Smith's farm. Very specific, while also not helpful at all for any reason. And so I definitely had my work cut out for me. So I started not really knowing what I was doing. I had never worked in collections before. I had never done anything with fossils beyond this one time in elementary school going on a birthday dig with a friend. So it was definitely a major learning experience. So I set up an Excel file, and throughout the time of working in the collections, I kept adding more stuff. So I wrote down where they were found in the collections, which drawer and which cabinet and all that stuff. The specimen number, if it had it, which is very important. The specimen name, if it's named. The collector, if named. How many specimens were in a box? Because occasionally, such as crinoids, they're in multiple pieces. The locality, such as 50 yards south of the large oak tree on Mrs. Smith's farm, or hopefully longitude and latitude, which is a lot more helpful. and then also our formation and member of rock unit. Along with that, I examined the boxes that the specimens were placed in, and I replaced broken or damaged specimen containers. An example, I was able to dig up my first draft of my collection. So we have cabinet, drawer, number, and then just all sorts of stuff I could find any information about any of these specimens on. This one was draft one. I got to about 2,000 different specimens before I had to kind of revamp it. And I did other stuff in the collections. I rewrote specimen tags. As I said earlier, a lot of these were written on newspaper, they were written on envelopes, they're written just with pencil in 1901. And so the paper was degrading, the ink they used wasn't acid-free, or it was just, written in 1901 cursive. And thankfully, due to work with my grandfather on his handwriting, I was able to read lots of different handwriting. And so, so I was able to use official University of Nebraska Museum specimen tags and rewrote it with acid-free museum pens. So I did it if it was damaged or faded. I rewrote it if it was on unofficial paper, and I rewrote it if it was in cursive or shorthand to make sure that everything was pretty well streamlined and pretty easy to understand if you're a graduate student just trying to get some data. And I also started noticing that we had specimens from the Peabody Museum. And we're not the Peabody Museum. So I started pulling them and placing them in a separate area so we could send them back and return them 50 years after we were supposed to. But they're back home and everybody's happy. So I stopped working in the collections when I graduated with my undergrad because it was time for me to start focusing on my master's. And however, the work is not finished, so they're still working. A retired professor is currently working on my Excel my Excel spreadsheet, adding more specimens, reorganizing the collections, just making it a lot more user-friendly. Along with that, we have echinoderm ammonoid and trilobite research currently being undertaken in our collections, along with being written up. And we also gave talks at GSA. Some of the collections were used for GSA poster by an undergraduate. We have multiple talks at GSA, which is the Geological Society of America, along with NAPC, which just occurred a couple weeks ago, which is the North American Paleontological Conference, and along with multiple papers on the way to being published. But I was invited to give this talk, so I'm going to talk about me. So how did this collection work help me with my research and my growth? So it helped me understand the effort behind museums the public doesn't see. So I grew up in Lincoln. I spent years coming here with school and with friends and, looking at all these fun taxidermied animals, looking at the fossils, all that stuff. But I didn't recognize how much stuff is in Nebraska Hall or in the back. I just thought museums were for us. Well, it is, but I didn't realize how much more there were to museums. So doing this helped me recognize that. It helped me discover my love for specimen research. I really enjoyed, as much as I hated pulling open a drawer and seeing pieces of newspaper with just really vague stuff written on it. I really enjoyed the puzzle of figuring out where it was and what was important about it, and just looking at what was drawn from the ground and placed in our collections for ever, basically. And it also developed my specimen handling skills. I wasn't required to wear gloves or anything, but you know, I had to have clean hands. I had to make sure I didn't, I don't know, chuck it against the wall, and just make sure that they're in one piece once I'm done. Or if they're not in one piece, make sure that they stay together and just don't jumble up stuff. So such as proper handling, organization, along with storage. It's so easy to just throw a box into a drawer, but if you do that, then nobody can use it. It's not helpful. Along with that, I was able to get an undergraduate research grant through the UCARE program at UNL. I did my entire grant in the invertebrate collections. I opened every single drawer in that collection and pulled out every single ammonite I could find and measured them and examined how the shell shape changed over time. It didn't. And I was kind of devastated because I spent 20 hours a week all summer long digging through these specimens and it's the same. But then I realized that they didn't change over time, but they changed over locality, which I put in the back of my head and thought about later during my master's research. So working in the collections helped me get a grant. And then that grant helped me get into graduate school. And I was able to get a position as a research assistant in Pete Wagner's lab to continue working in the collections, along with working with R and all sorts of other stuff. This work in the collections and this work with the grant and all of this stuff allowed me to move forward with something new in my life, even though my undergrad degree was completely unrelated to what I'm currently doing. This work in the collections also allowed me to be accepted to be a visiting researcher at multiple museums, such as the American Museum of Natural History in New York City, the Smithsonian in DC, which I actually spent 2 weeks at, and the Field Museum in Chicago. Some of these specimens I was able to measure went from tiny, tiny, tiny to very large. So basically, at the end of the day, the collections work I did in undergrad, not only did I help the university reorganize and get set up so that it's more accessible for future students, it also laid the groundwork for my current work and my future work. With that, I'd like to say thank you to Morrill Hall. I'd like to say thank you to Scott and my grandparents. And I'd like to say thank you to everybody who has helped me along my path. With that, I'm done.
Any questions, locally? Yeah. Did you ever look up any biographical information on some of the collectors?
I looked up some of their papers occasionally. I kept running into Fabian or Fabian. I don't know how it's pronounced because I've only seen it written. And I ran into some of his stuff at other museums, which was really interesting. A lot of the stuff I ran into kept popping up later in other papers, so that it kind of really helped me mentally place myself in localities and time periods.
I think it was Roger Pabian.
Fabian?
Yeah.
Oh my gosh. He's terrible at writing his own name.
Hard to read.
Fabian.
Yeah. You must have been the rest of you.
Any other points? Yeah.
So, great talk, really enjoyed it. Do you have any plans with this location trend that you put in the back of your head?
Yes, it's my PhD research. So my undergrad pointed towards the idea that time didn't impact the ammonoids, but environment did. And so during my master's research, I've been doing phylogenies, which is why I met Scott. And I also optimized my phylogeny for location. And there's a very distinctive trend in an early break between ammonoids that lived in shallow water and ammonoids that lived in deep water. And there's a lot of my genera are very polyphyletic, but they group together in locality. So I'm going to be looking in my PhD into environment, such as localities. So were they in deep water? Were they in shallow water? Along with water temperature and what their prey were and all of that stuff to then look at what about the environment is impacting their shell shape? What about the environment is impacting how their sutures, that are the squiggly lines on the shell, what about it is impacting those specimens? Because it's leaning less towards phylogenetically informative and more environmentally informative.
The cool thing about that is these are 500 million.
50 million.
They're 50 million years old. It's not like we can just go out and collect some things here. And so it's so cool. So that's the nice thing about this. So any other questions? Anything out on the internet? No?
Thank
Okay,
you.
good. So, well, thank you everyone for being here for this amazing ammonoid talk, or kind of ammonoid talk, collections organization talk, which is really very interesting because this is what we do all the time. So, all right, thank you.
Thanks.
All right. Really cool. Congratulations on getting done next week. I think I have to read it. We'll see how it goes. But you know, in general, it works well, right? I mean, she gives me stuff, I read it, I give it back, and it's good. We've done that a few times now. So Lynn was in my phylogenetic systematics class, so that's how I got into this stuff. Let's see here. We're now going to. Are you next, Abigail? So let me see if I can pull up your talk. Did I put it over here on? I hate this new Windows thing. Move this out of the way. Come on up and help me figure out what I'm supposed to be starting. Where did I put it? I thought.
I saw it earlier when you were pulling.
Oh yeah, maybe it's under.
in the download.
Yeah, let me go there. Come on. There's Carl. Hi, Carl.
Hi.
What's it called?
It should be my.
Yeah, I think it's right here under download. There it is. Yeah. Yeah, that's it. And we'll start. Yeah. So I push this button here and push this button there. Then I can go here. As soon as we're done with this, I'll have this all figured out. Yeah. Oh, and it's all going to be different because it's a new operating system.
How do you pull up the little laser?
Yeah, here. We'll start it right now. And then you go here, and then go laser pointer here. And then you got it. And then so we'll click here, and then you can use the arrow keys.
Perfect.
Just right in line. So Abigail was one of our top students in field parasitology, but she also took my course. She took field parasitology and parasitology at the University of Nebraska when I was teaching, or I do teach those courses. And so she showed out at Cedar Point Biological Station, wondering what to do. And I said, I have a project that's been ongoing for 5 years. Let's continue working on. And so she jumped in on it and she's still working on it, and maybe we'll publish it before she finishes her veterinary degree, which she starts this fall here at the University of Nebraska. So we're really lucky that she's staying nearby because obviously she'll be able to come into the lab all the time.
What am I studying?
Yes. Okay, so thank you very much.
Of course, of course. Okay. As Scott said, my name is Abigail Horner. I've worked in the Mantur lab. I've worked in the Mantur lab of parasitology as an undergrad, and I've studied the parasitic mites of back swimmers of western Nebraska. So I'm going to go into a background of the two main species I've been studying, as well as the results from the studies during my parasitology class and my systematic taxonomy class. So our parasite host is Notonecta undulata, common water boatman. It's the only species that Hydrachnophyllotera, the mite that I am interested in studying, has been observed to parasitize. It inhabits small ponds, lakes, and semi-permanent waterways, and it disperses by flying amongst water sources like cattle tanks, semi-permanent streams, and waterways. This is our parasite in question, Hydrachnophyllotera. It's in the process of being identified. The adult is free living, and the larva is identified by its Valentine heart shape. Other mites are just a plain round circle. And this is the primary method I use to identify. This is the wing of the water boatman. And the mite attaches to the underside of the wing and sucks on the endolymph until it becomes a nymphochrysalis. So it's just been engorged by feeding on the endolymph. Let me bring out my pointer here. These two on the bottom are a different species of Hydrachna, and this is the species of interest, Phylotera, up on the top. So it looks a little bit different, even in the nymphochrysalis stage. This project was originally started by Gretchen Lusso as a student in field parasitology in 2018. She originally identified it as the genus Hydrachna, and part of my research is making sure that it is indeed a water mite that's part of the Hydrachna genus. She took a hiatus during 2020 due to the COVID-19 shutdown, but in summer of 2022, I picked up her research, and I wanted to determine first, before going into any phylogenetic research, I wanted to determine if there was a difference in populations of Hydrachnophyllotera between different locations around Cedar Point. So I collected 600 of the common water boatmen, 200 from 3 different locations, and I used a dissecting microscope and forceps to determine if there are any mites, and if so, do I have the mite of question, the Hydrachnophyllotera. This is a map of all 3 locations that I collected from. For some semblance of scale, between the North Cedar Point location, which is a cattle tank, and Beckius Pond is about 2 miles. So all the way out to Haythorne was about 10. After all is said and done, and I identified all the mites, I calculated the prevalence of infection from these specimens. We found a 41% infection of Hydrachnophyllotera at Haythorne cattle tank, 32% in Beckius Pond, and 16% at North Cedar Point. I took the number of mites found at each of these 3 locations and used an ANOVA test to see if these findings were statistically significant, and our p-value was below 0.05. We found these very significant. I also conducted a post hoc Turkey test to determine pairwise if there was a significant difference between the pairs. The most interesting that we saw was a low p-value of 0.003 between Haythorne and North Cedar Point cattle tank. We had some hypotheses on why this is. We looked through literature, and we did some general observations such as predation, salinity, but we haven't found anything to back up why these are so different from each other. This is the life cycle of Hydrachna and Elias, described by Smith in 1977. So Hydrachna starts out as an egg at the bottom of a lake or a pond,
and then they emerge into pre-larva. But once they attach to a host, they become larvae, and they go into a nymphochrysalis stage, which we saw earlier, emerge into a deuteronymph, in which it detaches from the host, goes into a telechrysalis phase, and then eventually into an adult. For the Mantra Lab, we are able to collect several specimens from these 3 different locations of the adult, the larvae, and the nymphochrysalis, as well as the deuteronymph. I was able to, from what I believe, observe a telechrysalis, but unfortunately I was not able to collect any for the museum. And then we have not been able to observe the egg or the pre-larva stage. So in my systematics course in the spring of 2023, I then proceeded to create a phylogenetic tree for Hydracna. So the original plan was to use Hydracna phylloterra in this analysis, but the live specimens that we collected to propagate here in captivity so we could see the full life cycle, they were not viable. So we decided to create a tree of Hydracna, and when we get the data that we need from the live specimens, we're able to plug this new data into this pre-existing tree and get a better idea where Phylloterra falls in Hydracna, if at all. So I selected 6 different species from North America to serve as the in-group, and I obtained data from the literature about measurements of mouth parts, setae, a lot of small, minute measurements. And I selected 4 other possible species for the outgroup. We estimated the evolutionary and phylogenetic relationships using TNT version 1.6. And then as a test of goodness of fit, we conducted a bootstrap analysis of 1,000 replications with the shortest, most parsimonious tree. And we compared the percentages to a 70% threshold. That means that 70% of the time, and out of these 1,000 replications, that 70% that this branch occurred. So it's a more consistent tree. So this is the final tree that we came up with. The geographic labels are where each holotype was collected. The holotype meaning the original specimen we used to identify. So we use Arrhenius japonicus as the, Japans is the shorthand, as the outgroup, because it produced 5 distinct branches, all above the 70% threshold for the bootstrap analysis, and Berrii and Severensis were not in a polytomy together, and it gives us a more specific outlook on where they fall in the phylogenetic tree. All the other outgroups weren't very specific, and we would see a lot of tight grouping of the branches, so we wouldn't be able to identify where in the tree they would fall. Conclusions and plans for further research. So this systematic analysis is a great start. We want to start next by observing the full life cycle of all the species included. And we'll need more morphological analysis from the specimens that we've collected here at Mantor. Additional collecting is needed all of North America for morphological, molecular, behavioral, and other possible data such as parasites and viruses of these kinds. These are my references. And thank you for Dr. Gardner for teaching me during these classes, being my mentor, the Whaley Heading Society for producing TNT, the method used to create that phylogenetic tree, and all the students that helped me collect specimens.
Very cool. So this is an example of a pure undergraduate work, just like, kind of like Allison did, but Allison had actually already finished, but this is while Abigail working undergrad, she got to all this stuff. So we have a lot more to do, of course, but there's not enough time, it seems, when they're an undergrad to actually do all the work that needs to be done. So any questions for Abigail from out on the internet or here? So I think what that shows, oh, go ahead.
Yeah.
Maybe it's just because I don't take care of my aquarium. It's easier to do balls than it is to do water. So I think with that tree pointed to, we didn't show you all the trees that were available. What it indicates is we need more data. So swapping in the outgroup indicates changes indicating we need more data. Basically, we'll have to probably go to molecular methods to get more data. So, one gene will be one data point. So we have to get lots of genes. So, anything else? Anything else from anyone? So, we're having basically level first, which is, we have a little time for discussion if anybody wants to yell over the kids. If you'd like to talk about more things right now, if not, we can, shoot on over to, Nebraska Hall for lunch. So I can find out whether it's been ordered. I'm not sure. So, I will not pick up lunch and then meet everyone in Nebraska Hall. Thank you so much. And then, of course, tomorrow morning we'll reconvene here at 9 o'clock for the climate change. Climate discussion.